Hey everybody, welcome to week 6 live stream. Today we're going to be talking about cell signaling and the cell cycle. So if you guys have your worksheets, please feel free to do it along with me, and I am going to go ahead and get, yes, I'm going to go ahead and get started. Okay. So how are signals translated into cellular responses? Today we're going to be specifically talking about the G protein linked receptors, the receptor tyrosine kinase, and then ion channel receptors. So all of those are plasma membrane proteins, which means that they're protein receptors that are found in the cytoplasm or nucleus of the cell. Oh, sorry. So these kinds of receptors are in the plasma membrane, but then there are other protein receptors, of course, that are found in the cytoplasm, nucleus, and those are a different type. Some signals, for example, protein hormones, interact with signal receptors in the cell membrane to initiate the process of signal transduction. This often involves changes in a series of different relay molecules in a signal transduction pathway. Ultimately, the transduced signal initiates an intracellular response. Other types of signals, for example, steroid hormones, can diffuse through the cell membrane and interact with intracellular receptors. Examples of this are testosterone that interacts with its receptor in the cell cytoplasm, enters the nucleus, and then causes the transcription of specific genes. For G proteins and receptor tyrosine kinase system, the first question is going to ask how these two are similar. And we are to consider the structural similarities as well as how they function. So in both systems, they are bound to the cell membrane. And that the binding of a signal activates them. Once activated, these proteins interact with relay proteins and activate those. So once a signal reaches this protein receptor, that then activates relay proteins to go and spread the signal further. So I'll just go ahead and write that down. Interact and activate relay proteins. So an example of this for G protein specifically, and yes, this is recorded. All of these live streams will be posted to the Learning Center YouTube channel if you'd like to rewatch. But an example could be cyclic AMP, so that's kind of that signal transduction pathway. And then activated relay proteins. Oh, sorry. Sorry, everybody. Uh-oh, deleted some. Activated. So first activate relay. Activated relay. Triggers cellular response. And then the second question is asking where they differ. Okay, this is odd. So I can see everybody's messages, and I actually have my Streamlabs open on a different PC than I do my Twitch, and so when I'm watching the Twitch I can see just fine. I have been in contact with the professors about this, and I am trying my best to fix this issue. I just am not sure how to do it on my end. Okay, but the differences. So first difference is that okay. This is so strange. Okay, I'm gonna go ahead and keep going. If you guys can see the chat, then I am gonna post the extra credit link so you can get credit for that, but make sure to just watch the recording, and I apologize for the technical difficulties that continue to happen. So for number 2, one main difference is that the G protein linked receptor is a single unit, so G protein is one unit. And the 2 tyrosine kinase receptor proteins are separate, but when they're activated by signal molecules, they aggregate and become active. Has 2 parts and is joined together when activated. One other difference is that the activated G protein linked receptor activates the G protein, which is membrane bound, and converts GDP to GTP. This then moves along the membrane and activates a specific membrane-bound enzyme which produces the cellular response. But in tyrosine kinase receptor, they aggregate and can activate up to 10 different relay proteins. So that can have a different transduction pathway and produce different cellular responses. Is associated with GDP to GTP and activates specific enzyme tyrosine kinase, I'm just going to put TK, can activate um, 10 different responses. Okay. So they both are bound to the cell membrane. They both become activated by the binding of a signal, and they both activate relays which triggers a cellular response. The difference is that G protein is one unit, tyrosine kinase has 2 parts that come together once they're activated, and the G protein has a more specified enzyme which it activates, whereas the tyrosine kinase can activate multiple different responses. Number 3. Both systems can generate elaborate multi-step signal transduction pathways. These pathways can greatly amplify the cell's response to a signal. The more steps in the pathway, the greater the amplification of the signal, and explain how this amplification can occur. So in a signal transduction pathway, each activated enzyme or second messenger has a potential to catalyze more than just one reaction. So each of its reaction products similarly has the potential to trigger more than one reaction, and so the effects produced by that single molecule is greatly amplified. And I'll write that down here. So activated enzyme and second messenger. And yes, there will be a Google survey. I'm going to be doing a Google survey every live stream, just so I can make sure that I have you guys all down accounted for each week. And then when you guys go to get your extra credit, I will cross-reference, check, make sure you came to 5, and then I'll give that information to your professors. So one signal is greatly amplified. Also, the bloop juice, which is hilarious. Will you just write your name and your PDX email in the chat, and I can get you on my Google list? That would be great. Okay, great. So that was cell signaling. We're now going to move on to replication within the cells. This mainly, there's two names, mitosis and meiosis, and though they seem pretty similar, there are some key differences. And so we're going to go over that now, first starting with mitosis. So the overall purpose of mitosis is to produce daughter cells that are exactly like the parent copy. Oh, not how you spell. So first, the cells first duplicate their chromosomes, and then the chromosomes are equally divided amongst the daughter cells, and each has the same number and kind of chromosome. Awesome. So using Drosophila oh my gosh, I'm always bad at saying this, melanogaster, I just call it Drosophila, has a total of 8 chromosomes, 4 pairs, in each of its somatic cells. Somatic cells are all cells of the body, except those that will divide to form the gametes, ova, or sperm. So somatic cells are pretty much everything else than what we use to reproduce. Keep in mind that the stages of the cell division were first recognized from an examination of fixed slides of tissues undergoing division. And on fixed slides, cells are captured or frozen at particular points in the division cycle. Using these static slides, early microscopists identified specific arrangements or patterns of chromosomes that occurred at various stages of the cycle and gave these stages names: interphase, prophase, so on. Later work using time-lapse photography made it clear that mitosis is a continuous process. Once division begins, the chromosomes move fluidly from one phase to the next. So this is just having us basically look at what is happening with mitosis. So we have this diagram. Nobody can hear me? Okay. So for those who can't stay the whole time, I'm also just putting that link in the chat. I'll probably do that a couple of times just to make sure everybody gets it, and so those who need to access it a bit earlier can. Great. Okay, so this is mitosis. We see prophase, prometaphase, metaphase, anaphase, telophase, cytokinesis, and then the resulting daughter cells. So what we're seeing here, each of these chromosomes in prophase are being duplicated. And this switch over to prometaphase, we can see this nuclear envelope dissolving, and these spindle fibers are starting to now attach to the centromeres of those duplicated chromosomes. At metaphase, the duplicated chromosomes that are attached to their spindle fibers line up at the metaphase plate. That's why it's called the metaphase plate. That's a good way to remember that. It's not so intuitive, but there you go. Once these are all lined up, they're attached to their spindle fibers, each one has its own, then they're going to get pulled this direction toward opposite sides of the poles of the cell. So you can kind of see a preforming of what our two daughter cells are going to contain. Once we move from anaphase to telophase, these nuclear envelopes are starting to reform. The DNA is now not looking as condensed. And yes, you're going to put your instructor in the instructor box. I didn't do that last week, but it's just so I can keep you guys organized by class. And then during cytokinesis, now we're seeing the cell actually splitting. They're forming into two. And then the result, of course, is two daughter cells. We can't see the DNA as clearly as we used to. That's how we know it's safe and sound. And these daughter cells are going to be exactly like their parent. And I will probably be referring to this image a few times throughout the next couple questions. It asks how many cells are produced at the end of a single mitotic division. You just put your own instructor that you're taking for the general biology series. So whoever is your instructor for Bio 211 right now, you can put that in that box. Great, so two cells are produced. And then how many kinds of cells are produced at the end of a single mitotic division? There is only one kind. So we do have two cells. However, because I am kind, but because they are exactly like their parent, they are going to be just one kind of cell. If you're starting with a somatic cell that has, all like the chromosomes code for, I don't know, blue fingernails or something, and then you divide it, you're just going to have that same gamete in there. Great. So then in this table, it's going to ask us to deduce the chromosomes that we have based on the centromeres that we see. So we know that we are used to seeing chromosomes that look like this, with this guy here, but we know that in each individual one it has its own centromere. And so when it says 6 centromeres are observed in a prophase cell, then the pairs of chromosomes are 3 pairs. And then for each stage of mitosis, indicate the number of centromeres you would expect to find and the number of copies of chromosomes attached to each centromere. Prophase is when we're getting ready. So there should only be one question about instructor where you fill in their name. And so, okay, so for prophase, we're getting ready. We're starting to prepare for division, and so we have to duplicate. And so the number of centromeres visible per cell, and prophase we haven't divided yet, and we're also duplicating the chromosome. So we're still going to see 6 centromeres in prophase because we haven't really done anything yet other than preparing the cell for their next steps. So the number of chromosomes, copies attached to each centromere would be 2. That's when we're going to see this guy versus this guy. And then in anaphase, which we saw back here, now that they're split, they're no longer like this. So let's say we had 6 chromosomes, right, like we're saying. Once anaphase pulls them apart, they are going to have their own centromere. If the chromosomes do not have a centromere, division cannot occur because there's nothing for the spindle fibers to attach to, and that DNA will be just degraded and not used in the cell. So for anaphase, we're actually going to see a doubling of those centromeres because we're pulling them apart. If you can see here, it almost looks like we've doubled this to this, and that's why it would go from 6 to 12. However, the number of chromosome copies attached to each centromere now is 1. Awesome. Now we're moving on to meiosis. So this specifically has to do with, you can say sex or gametes or reproduction. I'm just going to do reproductive gametes. And so the overall purpose, I'm going to put the form in just one more time, scatter it throughout. The purpose of meiosis is to reduce diploid, which is 2. You can see di is always going to tell you that there are 2. Diploid chromosome to haploid, which means we're just going to have this diploid. And the important thing to remember is that this is a very specific part of the chromosome. So when we're dividing that diploid chromosome to a haploid, that it is a very specific half of the chromosome that is in the haploid. It's no longer a somatic cell where you can assume everything is the same as its parent, but there are specific differences in meiosis where you're going to produce 4, but those each 4 are going to be unique from the parent. The box for question 2 is on the next page, but I'll just go ahead and read it. It says, what types of organisms and cells, in what types of organisms and cells does meiosis occur? And we said that it occurs in the reproductive cells, but also specifically in eukaryotes. I'm having a hard time spelling today. And eukaryotes specifically that produce gametes. Which is ova or sperm. Awesome. So this next diagram is an overview of meiosis now. So looking here, I know that it looks a bit much, and I'm going to try to make this a lot bigger for you guys. Okay. I like to think of this in half. So we see prophase 1, prometaphase 1, 1, 1 over here, then we see 2. So there's going to be 2 cycles in meiosis. Prophase 1 is similar to the prophase in mitosis, where it is getting ready for cell division. You're going to see the duplication of chromosomes. The only difference is that in meiosis, something called synapse occurs, which is crossing over. When 2 duplicated genes cross over and they complement each other, they can swap genetic information and make that chromosome unique. And similarly, we see that duplication, we see those spindle fibers attaching, we see lining up of the metaphase plate, and we see anaphase pulling it apart. However, if you've noticed, the big difference is that in telophase, we're still seeing these chromosomes together. They don't look as they did back in meiosis in telophase. Do you see how in telophase, the chromosomes are looking like this, they're getting back to their original shape, they're doing their thing. But in the first part of meiosis, they stay this way because they have 2 other rounds to go to. So you see the reforming of the nuclear envelope, cytokinesis splits those cells in 2. We have the 2 daughter cells, but a sperm or egg cell goes through meiosis is correct. Just 1 second. And a zygote is what happens when 2 haploid gametes fuse. So the zygote doesn't go through meiosis. The sperm and egg cell undergo meiosis in our own reproductive cells. But when 2 haploids fuse, they produce a zygote that's unique. Great clarifying question. Thank you for asking that. So we're seeing these chromosomes still doing their little X thing at the daughter cells, and now they're moving on to prophase 2. So things basically happen once more, but things are being divided further. So we're just going to take one of these cells. We're only looking at one, but you can assume this same thing happens for both. So this daughter cell is also going through it. We have the attaching of the spindle fibers here. They're lining up at the metaphase plate. Anaphase is pulling those suckers apart, and then we're seeing a more familiar telophase that we did in mitosis, where these chromosomes are now looking less condensed, they're looking more like their natural state, cytokinesis can occur, and daughter cells are produced. Remember, the DNA, the genetic information in these cells are unique due to synapse or crossing over. Here, let me see if I can switch over. I'm using the mic on my computer that's attached to my webcam, so let me try to put on my headphones instead. Hello, hello. Does that sound better? I hope it helps somewhat. And so, to answer your question about one daughter cell from a sperm cell and an egg cell can fertilize, yes. So these daughter cells from this meiotic division are able to undergo the next process and form a zygote. So these can replicate, and they're doing it on their own. And then one of these daughter cells is going to come out, meet its mate, and then they're going to produce a zygote. Awesome. Great. Okay, awesome. Yay. I'm happy it's making sense. Yeah, of course. Please ask any questions. Use my time. I'm here for you guys. I went through the general biology series last year, so I totally understand how this material can be challenging, and I definitely want to help you through it. Okay. I'm putting the form in one more time, just moving this around. Okay, now we're looking at the products of meiosis. So A, how many cells are produced at the end? So one cell produces 4 daughter cells. How many chromosomes and which chromosomes does each of the daughter cells contain? Each daughter contains half the number chromosomes than the parental. It's also important to note here that each daughter contains one member of each pair of chromosome in the parent. And yes, so each daughter cell is a haploid for the gametes. The daughter cells produced by meiosis are haploid cells. Moving on to the table, if 6 centromeres are observed in Prophase 1 cell from another species of insect, how many pairs of chromosomes does this organism contain? This is similar to mitosis, as we said, so this is just going to be 3 pairs. Awesome. Now, in Anaphase 1 and Prophase 2, we want to know the number of chromosomes visible per cell and the number of chromosome copies attached to each centromere. So I'm just going to go back to our diagram here because we're looking at Anaphase 1 and Prophase 2. So in Anaphase 1, the number of centromeres visible, remember we're still in the first. We have 3 pairs of chromosomes, 6 centromeres, they're attached at the metaphase plate. Anaphase is now pulling them apart. We're going to have 6 still, as we did in mitosis, and the number of chromosome copies attached to each centromere is going to be 2. However, in Prophase 2, that daughter cell from the first division in meiosis is going through it once more. So the number of centromeres visible per cell in Prophase 2, the second part of this, is going to be half the number that we saw before, the common theme in meiosis. So we're going to see 3 here, but interestingly, the number of chromosome copies attached to that centromere is going to stay the same. And we can see that here. Now there's a lot of writing. Sorry, it's getting a little muddy. Okay. Non-disjunction of sex chromosomes. So non-disjunction is when they don't divide as we intended them to. So maybe one division gets one more chromosome, the other one doesn't actually get anything. That's what non-disjunction is that we want 4 haploid daughter cells, but sometimes things can get a little funky in division and that number does not always come out perfectly. So we're going to answer some true or falses to see what is likely and what can or cannot occur during division. So we see non-disjunction of sex chromosomes during human gamete formation may lead to individuals with sex chromosome trisomy. That just means that instead of an XX, which is 2, we're seeing XXY or XYY, things like that. An individual with the sex chromosome of trisomy XXY may have resulted from non-disjunction occurring in Meiosis One. And do you guys think that Meiosis One in the father's sperm production could produce triploid? True or false? I just want to see what you guys think. Yeah, so it is true. So if we're looking back at Meiosis One, the sperm production will result in some gametes with both X and Y, and then maybe some with neither an X or a Y. So if we are going to, and I'll just erase some of this so that we can draw this question out a little bit more clearly. So if 4 cells are produced, oh, wait a minute, I'm so sorry. We're talking about Meiosis One. I'm getting a little ahead of myself. So some gametes could have both X and Y. Some could have neither. And so if this went on to fertilize with an X, then the daughter cell that would be produced would look like this instead. This is a Y, because these were not able to separate. And that's kind of a tricky question. Not that I don't want to answer, is it from crossover? Crossover generally just has to do with genetic swapping, and it is true. If any of you are pre-med going into genetics and things like that, you'll learn a lot more about how crossover can affect this. And so the short answer is yes, it can come from crossover, but there are also other things in division that can do this. If a crossover happens at a certain point on a chromosome and not the other, some genetic information might be left out, some may be duplicated, things like that, and that's a whole nother rabbit hole. But for this case, just the division in Meiosis One, sperm production could result in gametes that have X and Y or neither, and so those that are with X and Y can produce trisomy, so that would be true. Some with X and Y, some with neither. So for the next one, Meiosis 2 in the father's sperm production, this one would actually be false. The reason for that is because non-disjunction during Meiosis 2 of sperm, because sperm is specific because it's XY. Ova have XX, so non-disjunction can happen at multiple times, but because it's 2 specific different chromosomes, it's not as likely to happen in Meiosis 2. So if non-disjunction were to occur, then it would either produce something with 2 X's or 2 Y's, or no X or no Y. So you can't have up here that some have XY and some have neither. You can only have in Meiosis 2, some have XX, some XY, because we've already gone through one division. We've already separated some of it. So once you separate X and Y, sorry guys, my spelling. I'm getting all my X's and Y's mixed up. But since we've separated X and Y, if you're going through Meiosis 2, that one cell has X's, that other cell has Y. They're not going to come in contact with each other once again when they're in 2 different places. So it could not produce an XXY individual, making 2 false. And then for 3, now we're looking at meiosis 1 in the mother's egg production. And we can say, like you guys brilliantly answered earlier, that this is true for pretty much the same reason that we said for the father's sperm. So the non-disjunction in meiosis 1 of egg production could produce eggs with 2 X's or no X. So you could have this in one cell or nothing. So if this were to mate with a Y, then you would get that trisomy that we're looking at. For number 4, meiosis 2 in the mother's egg production, like we said earlier as well, it's kind of the opposite that we gave for the answer. It would actually be true, and this is because meiosis 2 of egg production can still produce those with 2 X's or no X. They're no longer separated as they were with X and Y because you have 2 of the same chromosome. I mean, of course they're different, but you know, you have 2 X's. So you can still have an XX or nothing in your cell. And this can go through that same thing that we saw up here. So that's a very specific difference between ova and sperm, is just where non-disjunction can occur and how it can occur in meiosis. So there are specific rules when it comes to XX and XY, which make meiosis definitely more challenging to grasp, I think, than mitosis. So now that we've gone through 2 diagrams, we've looked at mitosis. We've gone through those steps. We've talked about crossing over. That the daughter cells are exactly the same as their parent cells. We produce 2 daughter cells in meiosis. There are 2 sequences of events that produce 4 haploid daughter cells. So now, in the next few questions, we're going to compare the 2, and I think this question 4 is true. Sure. So in meiosis 2 in the mother's egg production, so the reason why we said false for males is because once we've gotten to that second part, X and Y are in their own little cells, and so non-disjunction can't occur between these 2. You're not going to cross over. This cell either needs... Sorry, I think I'm not... I'm going to try again. I'm going to start over. So, in meiosis 1, we have the ability for non-disjunction in males to happen where there's X, Y, or nothing. For meiosis 2, they're separated. So you can have X over here and Y over here. They can't cross. There's no disjunction that's going to happen when they're separated. For ova with 2 X's, this separation is not as important because they're still both the same. So if you're separating these 2 X's here, non-disjunction can still occur because it's the same X in both. You're not dealing with 2 different chromosomes. Does that make sense? So we're seeing here meiosis 2. Yeah, so this would be the, if we're going to use this diagram, I think this could be super helpful. Meiosis 1, this is what they said we could have because there's non-disjunction. This cell didn't get anything, this cell got both. Up here, it can't happen again for men, for those with sperm. But when you have XX here and then XX here, it can happen once more. Non-disjunction can happen again because when you're separating these daughter cells, this one can have XX, this one can't. So I hope that makes more sense. Thank you, guys, for helping each other in the chat, too. Okay, so this has to do with the similarities that we see in this first part. The part of interphase for both mitosis and meiosis is actually identical. Both of them condense the chromosomes, both of them duplicate genetic information, and both of them get prepared for division. In prophase, we see that chromosomes coil in both, and condense. And yes, yeah, that is exactly right. Yes. Awesome. Okay. Yeah, of course. I hope if I know sometimes it can be hard for me to explain things in a relatively straightforward fashion, so I also appreciate you guys for bearing with me. The spindle forms. And the membrane breaks down. These are small boxes. Okay, the similarities in metaphase is that they line up, the chromosomes line up on the equator of the plate. On metaphase plate. The similarities in anaphase is that something always gets moved to the opposite poles by the spindles. Two op ends by spindle fibers. And in telophase, in both, the nuclear membrane reforms. And spindle breaks down. The differences are a little bit greater than the similarities. The similarities are definitely important, but the differences can also be just as important when it comes to exam questions. Just understanding the fundamental differences, I think, will be more beneficial than knowing all of the things that they have in common. So we know that there are 2 rounds in meiosis. A lot of these differences are honestly through meiosis. Mitosis is a bit simpler of a function. Meiosis has a lot of stuff going on. In interphase 2 of meiosis, there is no replication. No DNA replication. Prophase. In prophase 1 of meiosis, synapse occurs, or synapse. This does not occur in meiotic division. It's purely in meiosis that synapse and crossing over actually occurs. All right, so we know that in metaphase, there's not a bunch different. Both of them line up at the metaphase plate in order to be separated. But in metaphase 1 of meiosis, synapsed pairs line up, and in metaphase 2, individual chromosomes line up. Metaphase 1, synapsed pairs. And metaphase 2, individual chromosomes line up. Awesome. Anaphase is definitely the one with the most differences, I would say. So in Anaphase 1, members of each oh my gosh. Homologous pair separates. So homologous, if this is one chromosome from the same pair, these two are the ones that are going to separate from each other. Anaphase 1. Separate. In Anaphase 2, sister chromatids now separate. Sister chromatids separate. And then the only difference in telophase is that chromosomes don't uncoil during telophase 1 of meiosis because they have to go through another round. And Telo 1. Alrighty, now we're going to look at the amount of DNA and just compare it through the different phases. So we're just going to keep track of what we're starting with, what we're ending with in mitosis and both cycles of meiosis. The amount of DNA in a somatic cell equals C during G1, which G1, remember, is gap phase 1. We have gap phase 1, S phase, gap phase 2. We have that whole cycle, and so gap phase 1 is what is really, that's all interphase, right? And so that's what we're specifying because that's where it gets duplicated. So in interphase, there's only one C in gap phase 1, but then once it's duplicated, there's going to be 2 C's, which is DNA, in gap phase 2. If we're moving on to prophase, we're going to still have that duplicated DNA. Metaphase, they're lining up, but we still have that duplicated DNA. Anaphase, they're split, but we're still in the same cell technically, so there's 2. And in telophase, we have 2 to start with while things are starting to rebuild. But then, cytokinesis turns 2C into just C. So, for Meiosis 1, we're going to see something similar that we did in interphase of mitosis, where we're going to see C in gap phase 1. And once it duplicates, we're going to see 2C in gap phase 2. Prophase, we still have those duplicated DNAs. We still have those in metaphase, but they're lined up. They're getting split apart, but they're in the same cell, so we're going to have the same amount. And telophase, like we said up here, while it's rebuilding its stuff, it's still the amount. However, cytokinesis reduces the amount to just C. Alright, Meiosis 2. There is no duplication that happens in Meiosis 2. We already accomplished that here. So there's going to be C in G1, and then that C is just going to remain the same thing in G2. There's no duplication. We're not getting 2 DNAs in this one. So we're going to move this C along. We have the same amount in prophase, metaphase in the same cell. We have it in anaphase and in telophase. However, because of cytokinesis, it's actually going to be half C. Awesome. Next question says, how do the similarities in prophase of mitosis and meiosis compare to the similarities in telophase of mitosis and meiosis? Prophase deals with, so if we can see here, we're seeing chromosomes getting ready in both, and in telophase in both, we're kind of seeing that be undone once again. The cell is now reforming. So telophase can be thought of as the opposite of prophase. What's done in telophase, the undoing of the nuclear envelope, the condensing of the chromosomes, all of those things are redone in telophase. So telophase builds a nuclear envelope, it uncoils the chromosomes, all of that good stuff. So what I'll write is that telophase in both is the opposite of prophase. What is done in prophase is undone in metaphase. Alrighty, last question. Awesome. At what stages does most of the differences among mitosis, Meiosis One, and Meiosis Two occur? And for what reasons do these differences exist? So the primary differences between mitosis and meiosis mainly occur in prophase 1, where synapse occurs. So synapse is what allows the members of homologous pairs, which are just same pairs that can line up to one another, they complement each other, to separate to opposite poles and reduce the chromosome number at the end of meiosis 1 to half of the original cell. Mitosis stays the same throughout, and so the main difference is, like, mitosis can be thought of as a simple sequential step, happens the same every time, same DNA, same everything. When reproduction is occurring, there's a reason why I have my mother's eyes, not my father's, or I have my dad's hair instead of my mom's, that sort of thing. It's because the gametes that are produced are specifically unique, and that has to do with synapse. So we can put synapse and prophase 1, and I know that I keep going between synapse and synapse. I've heard it both ways, and I can't make a decision. Synapse and prophase 1. This reduces the chromosome number at the end of meiosis 1 to half that of the original cell. The OG cell. Cool. I'm going to put the Google form in one more time, just to make sure everyone awesome. Okay. Synapse is the crossing. So when we're in prophase 1, chromosomes line up, and actually, this is a better. So synapse is not the splitting of the chromosomes at the plate. Synapse is what happens when two chromosomes line up that complement each other, and then, like, this genetic information and this genetic information can swap. To produce unique looking chromosomes. That's what synapse is. Yeah, so like these, yes, this only happens in meiosis. So these two can be think of, if we're looking at it at more of a microscopic cell stage, if one chromosome looks like this, homologous pairs mean that they are complementary. It's going to look like these are part of their own chromosomes, but they can line up because they complement, and then things like this can switch. And so now it's ATGG and TACC until rather than its original. So this only happens in meiosis. In mitosis, you're not going to get crossing over. You're not going to get any of this to happen because you only want to produce the exact same cell that you started out with. But for meiosis, you're producing unique cells. Crossing over occurs or swapping information, like, hey, give me the brown eye gene, I'll give you the blue eye gene, that sort of thing. Does that clarify? Okay, awesome. Is there, yes, in mitosis there can totally be mistakes and errors of copying, things like that. Non-disjunction, what we talked about. Hey, Bethany, so kind of, is there other things like a specific place where it's kind of confusing? No, so synapse does not happen when the zygote form. Zygote is here. I'm gonna go do like just a blank thing of paper. Okay. So let's say we have 4 haploid cells. No, we're gonna have one gamete and another gamete. This worksheet, so it should be posted on your D2L, and if it's not, I would talk to your professors. But the blank worksheets are posted, and then after the live streams, the keys are also posted. So let's say you have one chromosome, another chromosome. Both of them come from different ones. These are both haploid. There's just that one chromosome in there. A zygote is what happens when these two fuse to make a little baby. This is what a zygote is. These are the individual gametes. So we can say this is ova and this is sperm. Synapse happens way before a zygote is formed. They happen when these gametes are being produced. So when meiosis is occurring in prophase 1, homologous chromosomes can line up next to each other that look like this, and they can swap information. They can like, give me this, I'll give you that sort of thing. And so when you're producing, after the end of meiosis, when you have 4 haploid cells, each of them is going to have different genes in there. Yes, yeah. So homologous chromosomes, which means like chromosomes, they are similar to each other. They're going to get right up next to each other, and they're going to swap information, and each haploid is going to be completely unique with new DNA. This is what is sperm. This is what is an egg. And so synapse occurs in the first part of meiosis. It swaps genetic information, and then you get 4 distinct haploid cells. Those haploid cells confuse with another haploid cell to make a zygote. But synapse does not happen past prophase 1. Yeah, I know. It's kind of, well, sister chromatids too, they're like, they're attached to each other. This is like the worst chromosome I've ever seen. chromatids are just ones that are attached to each other. So this is these are sister, and then these lining up like this. These two are homologous chromosomes. So this is a sister chromatid. This is a homologous chromosome to this one. And so this one and this one can swap. I feel like if I did this in color, it would probably be better. So I'm going to draw one that's red. And one that's blue. And then these can like cross over like this and like swap their DNA here. So these two are homologous chromosomes. And then these little individual ones are sister chromatids. So sister chromatids are like a part of this one chromosome and this one chromosome. Like if this is one sister chromatid, another one. So that's like one chromosome. But yeah, this is still one chromosome. So this one chromosome has two sister chromatids. And one of their sister chromatids can meet with the other chromosome's sister chromatids and swap. So yes. Yeah. So this is, if we only had one, this would be a chromatid. Here. Sorry I keep clearing the page. I just feel, I hate when things get muddy. Okay, so this one is a chromatid. This, however, is a chromosome. They're called sister chromatids because they're sisters attached to each other. So it's not quite like one sister chromatid does, the other one doesn't. We're honestly just looking at them in their chromosome. So this is a chromosome, right? A homologous chromosome is another chromosome that looks exactly like this, but just lines up with this one well. They line up together. So it's not always, I wouldn't get too focused on the nitty-gritty of like which sister chromatid, blah, blah, blah specifically. It's more of the homologous chromosomes lining up. And I know that this is like 2D, which sucks, but like if this is the side view of a chromosome and like you can see his pair behind him, it could like line up with another chromosome like this, right? And there could be multiple swaps of DNA. Yes. Does this help? I know that like the terminology when it comes to chromosomes and cell division is it's challenging. Homologous means similar, yes. So it just means that they're lined up next to each other. They're similar enough that swapping is not going to cause any damage or anything. So it's just like two chromosomes that are pretty similar to each other line up and they can swap information. And then they swap their information, blah, 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 and then they go on to divide. And yes, chromatids are just the two parts of the chromosome. So this is a chromatid, this is a chromatid. Okay, awesome. I'm happy that's making more sense. Are there any other questions? Yeah, of course. Any questions that you guys have, I'm totally here to help you. Of course. Awesome. Okay, thanks guys. I'm going to go ahead and end the stream. I'll make sure that this gets posted to YouTube by tomorrow, and I hope you guys have a really lovely evening and enjoy your weekend. Bye guys.